Okay. There you go.
Now you can't even see it.
Let's pull up the actual slides.
Squeeze. Do you not need this? Should I go leave the room?
No, you don't have to leave the room. You might want to. Is that the side forward? Yeah. Okay, so this is my pleasure to introduce Sara Lipshutz today for her exit seminar. Sara started her career as an undergraduate at Swarthmore, which really fits her personality. She is very free-spirited and learned to write there, became an amazing writer. I still give her actually my stuff to read. She's such a good writer. And so I think Swarthmore was really the source of that. And then she went on to get an Ernst Mayr fellowship to go to Panama and to do work on Chicanas, actually. And that's where her love of them started. And I think she was the youngest recipient at that point of the Mayr Fellowship. So it's pretty cool, right out of undergrad. And then after that, she went on the market looking for a graduate school. And I was lucky enough to get her. She was looking at a lot of different schools. I was a starting faculty member. In fact, I hadn't even started. I just had a baby. And she tells me that when I interviewed her, I was actually nursing her, which I have no memory of. So she clearly is a very brave soul and decided to be my guinea pig and start with me at Tulane. And I just was Rob actually sent her, Rob Brumfield sent her my way. I think she applied first with Rob. And he said, oh, you like behavior? You should go talk to Liz. So thank you. That's one of the best gifts you've given me. So Sara has been a joy to have in my lab now for 6 years. And I want to talk a little bit about all of her accomplishments. So she has done an amazing job with her research. She is a very serious scientist. She works really hard on her presentation styles, how she presents herself. She's amazing. She's very mature in her work. And she has, because of that, has given more than, I don't even know how many talks she's given in the time of her graduate career. I think we're rounding 2025 and invited workshops, symposia all over the country. She's been invited to present her work on both Chicanas and on white-crowned sparrows. And she, in just the last year, has given 11 presentations at conferences. So she definitely gets out there, which is great. She's made a lot of connections. And in that time, she's also authored 6 publications. All of her chapters, except for one of her dissertation, so 3 of her 4 chapters have already been published, which makes my life easy and her committee's life easy, so thank you. She has 1 more that's in prep and will soon be submitted. And she's also, in her time, has just been really conscientious about getting the grants. Working on that. She day, night, I get text messages, 2 in the morning, she's working on a grant, I think. She's got more than 25 grants and fellowships in her time, including an NSF GRF, an NSF GROW, an NSF DDIG, National Geographic, missing one, missing another like 20, so a bunch of Smithsonian's, Myers, all sorts of things. So she's been very good. When she came in, I told her that she could work on anything she wanted if she funded herself. But if she didn't, she needed to work on my stuff. And so she took that very seriously, and she went out and very much funded herself. Sara is also, Sara is also a joy to have in the lab. She has been an incredible senior graduate student. She's really shown the other students in the lab how to be successful graduate students. She's supported them in writing their grants and their papers and helping them take graduate school seriously. And I really appreciate that.
I think that's a piece of fried chicken.
It is. And it was, Sara did most of her time at Tulane in New Orleans, and you can see that New Orleans had a really important role in her time as a graduate student. And I think it was very hard for her to leave, but I still feel blessed that she came with me up here on this move, even though it was for just 1 year to UT. And I think UT has really benefited too. Just in the last year, she's served on a number of committees. She's become a member of EEC and GRIEB. She's done a bunch of outreach. She's mentored 4 undergraduate students, helping them get summer fellowships. Really has contributed. And that's just on top of everything else she did at Tulane, which I won't even go into. But she's done an amazing amount of service and outreach, as can be seen here, making science fun for undergraduates as well as for students in Panama, where she's worked. And she's just been really a role model for me too, making sure that you have outreach as part of your career. So Sarah's also, because of this amazing record, she's been offered many postdoc positions now, and she just accepted one with this woman, Kim Rosval, on the left. And you can see there's a similar, I feel like they're already going to get along. There's a certain approach to field work, and I think it's going to be a really productive collaboration. I want to congratulate Sara on her voyage forward. Tell her I'm going to miss her. Okay, oh, and one last thing. Oh, this is either a party of celebration or commiseration tonight after her defense. And there'll be some takeout, but if you can bring sides or desserts or drinks, want to supplement, please do contact Sara about addresses. I need to find that out too.
I can tell everyone, but it's being broadcast on the internet. So.
Yes. That's why I did it. So there you go. Okay, and with that, I will let Sara actually give her presentation.
Should always clear out my desktop. Okay. So thanks everyone for coming here today. It's good to see a lot of new and old faces. And I think to start out, I actually have a lot of people to thank. So especially Liz, my advisor, but also my co-authors, Joanna and Ole, Isaac, Mike, and Matt Miller, everyone in the Dairyberry lab, people in this room, but also former postdocs in our lab, and my lab mate, Jenny Phillips. And then my new University of Tennessee committee, as well as our Green Tulane committee, plus lots of feedback from our various writing workshop groups and different labs that I've gotten to visit over the years of my PhD. I spent a lot of time kind of traveling Panama, New York, and Switzerland, and now to here from New Orleans. So there's been a lot of people on the way, and especially lab and field assistants who, without them, this work wouldn't be possible. It takes a lot of hands to catch jacanas. And my friend Stephanie for doing some great illustrations, and then of course family and friends. Okay, so like many of you guys, biologists in this room, I'm fascinated by the diversity of life around us. And something that really drives me is, how did all this diversity come about, and specifically, how is it maintained over time once it's formed? And so, for example, we can see these colorful anole dewlaps, or these really diverse sparrow songs, and each of them varies dramatically across closely related lineages. These signals play an important role in communication, both in the context of attracting mates and repelling competitors. And there's also numerous empirical studies that suggest that these signals are important for species recognition, such that individuals can use these signals to discriminate between closely related congeners. And so because of this, it's thought that sexual selection plays an important role in the process of speciation. And so for effective communication to occur, a receiver here has to be able to recognize the relevant information content of a signal. And any breakdown in that signal, both in the context here of mate choice or competition for mates, can reduce the likelihood of mating and therefore reduce gene flow. And so as signals diverge across populations in isolation, this reduces the likelihood that they might recognize each other as mates when they come back into contact. And I use hybrid zones as this natural laboratory for testing the role of reproductive isolating mechanisms like mating signals in the process of speciation. So how are we maintaining those species barriers? And this is really effective for someone in the context of a PhD, because these signals can diverge over thousands of years, but we can look at these hybrid zones in our time scale to test the species barriers. And so avian hybrid zone in particular are good for studying this role of mating signals in the speciation process, because in birds it's thought that pre-mating isolating mechanisms like song or plumage are more important than post-zygotic mechanisms, for example, hybrid and viability. And so in my talk, I'm interested in understanding divergent sexual signals and the behavior that's important in mating. And I ask how species barriers are maintained. Specifically, what are the behavioral drivers of hybridization and reproductive isolation in two different avian systems? And so in the first part of this talk, I explore male-male competition in the white-crowned sparrow. And I ask the question, does song act as a behavioral barrier to gene flow? And then in the second part of the talk, I look at the system of jacanas, and I ask, does female competition promote hybridization? And so for the first part of the talk, these are our two players. So over here we have Zonotrichia leucophrys nuttali, or the Nuttall's white-crowned sparrow. I'll refer to that as nuttali. And we have Zonotrichia leucophrys pugetensis, the Puget Sound white-crowned sparrow, which I'll refer to as pugetensis. And so these are both subspecies of white-crowned sparrow. And here's their geographic distribution. So pugetensis is found in the northwest of the United States along the coast, from California up into Washington, whereas nuttali is found more in central and southern California. And there's a putative hybrid zone described between the two subspecies based on allozymes and some intergradation of morphological characteristics like their molt, because pugetensis up here is migratory, whereas nuttali is resident. So there is that divergence in behavior. However, no one was able to split these two subspecies as genetically distinct. But the reason that they were so well studied for decades is because of this. So each of these is a dialect, which is discrete geographic variation in song. So at each locality that you can sample across California up into Washington, the localities have all of the males there singing that specific dialect. And these are spectrograms, so they're visual representations of sound. You can think of it on the x-axis as time and the y-axis as pitch. And so the question that was really driving a lot of scientists on this system was, do these dialects play a role in the speciation process? So this is cultural evolution. These birds are learning their song. And can these dialects be a force of a barrier to gene flow? But in this talk, I'm not going to talk to you about dialects as much. I'm actually more interested in, what about the two subspecies? Is song a reproductive isolating barrier between those two of them? So we can actually approach it from a larger scale rather than this controversial dialect scale. And the other thing we're going to do, instead of thinking about songs as visually the way that humans do, we're actually just going to take the different parameters of song, things like the length of the notes or the bandwidth or the rate of those notes, which we can quantify. And so we're less biased by our human observer visual, and actually compare these songs between the two subspecies, as well as their behavioral divergence and their genetic divergence. And so, as I said, we could ask the song question at the dialect boundary, but as a first step, we need to figure out if even between those two subspecies, song is acting as a behavioral barrier. And so I have a series of predictions for how we might test this. The first is that these two subspecies should form distinct genetic clusters. And so what we're looking here is just a predictive figure of a structure plot. Each vertical line is an individual, and we see here that, as predicted, nuttali should cluster separately from pugetensis, but all of those individuals should cluster with each other. We also predict that since we're using this putative hybrid zone to understand the role of mating barriers, that we actually should have a model of secondary contact is the strongest support of a bunch of different models of evolutionary history. So one other alternative could be just ongoing gene flow. But we predict that there was a period of isolation and then they came back together. So allowing that divergence in song, which we can then test as a barrier. We also predict that those songs should be divergent between the subspecies. So ignoring all of these cultural dialects, are there features of songs specific across all dialects but within a subspecies that we can use to split them? And so we predict that they should cluster separately here. We also predict behavioral isolation between the subspecies. So here, what we predict is that if we play male birds on their territory, their own subspecies song versus the other subspecies song, that they should respond more strongly to their own song than to the song of the other subspecies. So this indicates discrimination based on that signal. And in birds, we think of song as this keep-out signal. So any kind of divergence, both in the song and the behavioral response to that song, can indicate that if a male were to come into a territory from another population, and that individual holding that territory was discriminating between that new male song and the local subspecies-specific song, that male might have a hard time defending or getting that territory if the current male wasn't recognizing that song. So we use these territorial playback experiments as kind of a proxy for how an individual understands and discriminates against that song, because tests of females are quite difficult. And then finally, we've got these separate patterns of genetic and song and behavioral divergence, but to actually start inferring the processes that are connecting these different patterns, we can now start relating how the difference in song affects the genetic distance or vice versa. So we can't infer causation, but we can start looking at the correlations of these two. And also whether the differences in behavioral response are predicted by how different those songs are. So to jump into the data, we found, as predicted, that the subspecies did form two distinct populations. So what we did to get this information was that actually Liz and colleagues sampled across the hybrid zone at 17 different localities, took blood samples from individuals, and then we used something called genotyping by sequencing, basically genome-wide markers which can give us an idea of the genetic information of that individual. And with that, using structure, we split these two subspecies for the first time. So no one else was able to do this with such few markers, but we had around 6,000 SNPs, or single nucleotide polymorphisms. We also found that there was admixture. So what this is meaning here, these are putative hybrids, and so each individual here has ancestry both from Pugitensis and from Nuttallii, suggesting that there really might be a hybrid zone there. And then I also collaborated with Mike Hickerson and Isaac Overcast to test different models of evolutionary history for this system. And so we did find strongest support for a model of secondary contact, where the two subspecies diverged around 9,000 years ago, which actually really well aligns with the history of that area. So during the last glacial maxima in the Pleistocene, these two subspecies likely diverged in isolation in glacial refugia. And then as the glaciers receded and they were able to expand their territories and their distributions, they came back together in secondary contact and started interbreeding. And that was around 2,000 years ago, so quite recently. We also found that the songs are divergent between the subspecies. And so here I'll just play you an example of what a Pugitensis song sounds like. And then what a Nuttallii song sounds like. And you might notice some visual differences between them. Here's what a hybrid song can sound like. But I think what was really interesting that we did here was that by ignoring these different dialects as visual representations, we could then look at 14 different acoustic parameters. And so what we found was that the genetic subspecies identity was a significant predictor of the song structure. And when we did a discriminant function analysis, we found that the whistle length was the best to split those two. So here Nuttallii had on whole a longer whistle than Pugitensis. And we took all those 14 different acoustic parameters and summarized them with a principal components analysis to kind of get a couple different representations of what those song differences are. And so we could then map that across the hybrid zone. And what we found here for song PC1 was that in black are Nuttallii and in white are Pugitensis, and that the hybrid individuals shared their song PC1 features with Nuttallii. And when we looked at the genetic information of these, we found that actually the hybrids were more genetically similar to Nuttallii. So this could reflect a difference in our sampling or geographic distance, but it seemed here like the genetic similarity was explaining the song similarity. And then we asked whether song was a behavioral barrier. So to do this, we conducted territorial playback experiments. So we went out and found territorial males and then played them the song, both of their own subspecies and of the other subspecies, and measured a suite of different behaviors that indicate their discrimination. So how much they respond, and then we can compare their discrimination based on the difference between those two stimuli. And so things that we measured were how close that they approach the speaker, so average distance, their song rate, how many times they sang in response to that stimulus, and how many times they flew over the speaker. And so we did these in 4 different localities to represent outside of the hybrid zone and inside of the hybrid zone. And what we found is that, as predicted, individuals responded more strongly to their own song than to the song of the other subspecies. And so this is indicating, the slope of the line here indicates discrimination. And then we can also look at the height of these lines. And so for some reason, and we don't really know why, I'd be happy to hear what you guys think, the hybrids responded more strongly overall, although they still did discriminate significantly between their own and the other, if that song was different enough. And so what we're seeing here is that the slope of the hybrid lines are actually weaker than between the subspecies, and that is explained by song similarity. So because those individuals in the hybrid zone did share more of their song with Nuttali, they actually didn't discriminate as strongly between their songs and Nuttali songs, but they did between their songs and Pugitensis songs, because those Pugitensis songs were more different. So this is allowing us to start inferring some of those mechanisms underlying behavioral discrimination, and here we're finding its song divergence. And then we were also interested in seeing if any of these different song features that we looked at could tell us something about the process of speciation, this process of genetic differentiation. And so what we did find is that looking across all these different acoustic parameters, that one stood out. Any guesses? Well, it's right here, but it's the whistle length. And so that was exciting to us because we knew that it was already good in predicting the differences between the subspecies. But when we were testing it against kind of a null model of geographic distance, so just a pattern of isolation by distance, we found that the whistle length was a stronger predictor of genetic distance. And that was really interesting because other work, for example, by Dai Shizuka and his grad student Emily Hudson on different species of sparrow in the same genus, the golden crown sparrow, they found that nestlings, even before they start learning their song, respond more strongly to the whistle alone of their own species song. And so the whistle may have some role in innate song learning. And we are really excited to pursue this further as potentially there being a kind of genetic template for song learning. And the whistle might have something to do with it because it's that introductory note in the song. So it's the first thing that these individuals are using to communicate information. So to summarize this part, we found that as predicted, the subspecies did form genetically distinct clusters, but there was hybridization in between them. We found strongest support for a model of secondary contact that aligns with the Pleistocene glacial maxima, last glacial maxima hypothesis. We did find that genetic subspecies identity predicted song structure, specifically the length of that introductory whistle. And we found that behavioral isolation is strong between parental subspecies, but it weakens in the hybrid zone, and that's based on song similarity. And that genetic song and behavioral divergence are all associated with each other. So a lot of previous studies would look at different components of these or just describe the patterns, but not be able to tie all this information together in these relationships. So this was really exciting. And as kind of a summary, we think that song is an incomplete barrier to gene flow. So I worked on the white-crowned sparrows in the first part of my dissertation. There was a lot of great existing data to work with, and it gave me a lot of good experience in learning how to use bioinformatics and working with genetic data. But the sparrows kind of have this traditional perspective of sexual selection and the speciation process. So we think about mate choice and competition for mates. The females are doing the choosing, and the males are doing the competing. And typically in this model of thinking, we consider the females as the gatekeepers of the species. And so because of their higher investment in offspring, we can predict that they should avoid hybridization. That can be costly. And so they should discriminate more between their own species or lineage than another. In contrast, we expect males to mate more indiscriminately because their sperm are relatively cheap. And so they might more easily or more readily hybridize with other species. And so I showed with the white-crowned sparrows, though, that actually, in a competitive context, male song could promote reproductive isolation if that song is different enough. And it made me wonder about another kind of sexual selection, female competition. And so female competition is actually quite widespread in nature across a diversity of taxa, but it's kind of been underlooked. And to understand the role of female competition in hybridization and the process of speciation, I thought, well, let's explore a system in which female competition is a major determinant of mating success. And so in the second part of the talk, I'm going to use jacanas as my system and ask the question, does female competition work like male competition in terms of promoting hybridization or facilitating hybridization? And so our system are jacanas. They're tropical shorebirds, and they have this really interesting polyandrous mating system. So poly meaning many, androus meaning males, and we can see that here. So this is a harem that a female has, and it contains 4 male mates. And this is a classical polyandry, so she mates with them simultaneously, and then deposits her eggs into their nest, and those males all take the role of incubation and parental care. There's also a sexual dimorphism in these species. So even though their plumage and their facial ornamentation looks very similar, the females are much bigger, sometimes as much as twice the size of the males. And because of the female spends her time defending the territory and preventing predators from coming to eat those chicks or nearby females from taking over the males, while the males do all the parental care, it's also considered sex role reversal. I should also say that not every female gets to breed. So within the system, there are residents and floaters, and only the largest females are the ones that can defend and maintain territories on which they breed. And the same goes for males. And luckily enough, there are 2 species of jacanas that hybridize. So now we can combine our predictions of female competition in a hybrid zone context. And these 2 species are Jacana spinosa here, the northern jacana, which you can find from Mexico throughout Central America into Panama, and the wattle jacana, Jacana here, which you can find from Panama throughout South America. And there's a couple of features that we can use to phenotypically classify these. So Jacana spinosa has a yellow trilobate frontal shield and brown chestnut plumage on the back and the front. And then Jacana has this red bilobate frontal shield and red hanging wattles, which are kind of like a mustache, like a handlebar mustache. And the subspecies that occurs in Panama is all black, although throughout the rest of South America, it's also brown. And then hybrids kind of have intermediate characteristics of both. So they have a yellow frontal shield and kind of a reduced red wattle. And their plumage is also a mixture of brown and black. And as Liz mentioned, I started this project the year after graduating from Swarthmore before grad school, and I worked with Matt Miller at the Smithsonian Tropical Research Institute. And we worked on a paper together kind of as a first description of the hybrid zone. So what you're seeing here is a haplotype network for mitochondria. And this is just like around a 652 base pair region of the CO1. And what we're looking at, so all of these bluish purple colors are representing Jacana spinosa. And all of these red colors are representing Jacana. And in yellow here are the hybrids. And so what we're seeing is that these hybrid individuals, and we were at this point only classifying them phenotypically, these hybrid individuals all shared their mitochondrial haplotypes with Jacana spinosa. But we didn't find the reverse pattern of any phenotypic hybrids sharing their mitochondrial haplotype with Jacana. And so knowing that mitochondrial DNA is inherited from the mother, one prediction that we had was, okay, so we're having these crosses of a Jacana spinosa female and a Jacana male. And given what we know about the mating system of jacanas, potentially these Jacana spinosa females are more dominant than Jacana females. And so when they come into contact in the hybrid zone, these females might be monopolizing mating and disproportionately mothering the hybrid offspring. An alternative, but not mutually exclusive hypothesis is that a cross between a Jacana female and a Jacana spinosa male isn't viable. So this project led me into some key questions in my dissertation, which is first, what is the genetic structure of the hybrid zone? We had some phenotypic ideas of what hybrids are and what the species are, but we had very few loci. And so I wanted to use genome-wide markers to understand the extent of hybridization between these two species and really how different they were. And then second, having these ideas about, well, there's potentially a bias in this inheritance of mitochondrial DNA. Could it be because of female competition? And so do the species females diverge in both their competitive morphology and their aggressive behavior? And then finally, given examples of what we've seen in other hybrid zones where male competition can actually facilitate integration that's asymmetric, so hybridization that for certain traits or alleles pushes them in one direction but not the other, might we also see that in females? And I'll explain this further. And so to answer these questions, I went out to Panama and Costa Rica. First, I actually looked on Google Maps and I said, where's the water? And then I said, okay, I'm gonna go and drive to those spots. So rented a car, got permission from lots and lots of really friendly farmers who let me stomp around on their farms looking for these birds. And we went all the way from the Darien up to where the Panama Canal, some of you may know Barrow, Colorado Island. So one of our sampling sites is here in Gamboa, all the way through the Azuero Peninsula, and then up into where we think the center of the hybrid zone is, and can confirm that now. And then even up into Palo Verde, Costa Rica, because we wanted to make sure that we were getting genetically pure Jacana spinosa. We weren't really sure at this point where the hybrid zone center was and if we had to leave Panama. So we did for one field season. And so again, I used genotyping by sequencing for around 271 individuals and heavily filtered around 13,000 SNPs across the genome. And so what we found, this is a principal components analysis of the genetic data, and we found that we were able to discriminate between those two based on PC1, which explained over 50% of the variation in the data, which is really high. And so we split Jacana spinosa here from Jacana over here, and then these were our hybrids in between. So the colors here, yellow is for Jacana spinosa and red for Jacana. And then the orange was our classification of hybrids. We also used structure admixture proportions for KO2. And so that's then how we classified the hybrids as anything between 0.1 and 0.9. And for genetic PC2, you're seeing here, well, it looks like it's explaining this variation in Jacana, and that's true. There's actually this pattern of isolation by distance in Jacana that we saw, but we didn't see it in Jacana spinosa, both based on a couple of different principal components of the genetic data, but also pairwise FSTs. And we actually could look a little further, and so not just that we have hybrids, but what kind of hybrids are they? And so there's a couple different ways to think about this. The first are the F1s, and so this is when purely parental Jacana spinosa and Jacana interbreed, and they produce this hybrid offspring. That's 50-50. But then we actually found evidence for back crossing. So that's when one of these F1 hybrids then reproduces with one of the parentals, and potentially there's even further back crossing. And so that's actually one of the most exciting findings here in this study, is because it shows this potential for integration. If we only found F1s, this hybrid zone might be a kind of evolutionary dead end. But knowing that these hybrids are able to then interbreed with the parentals, this allows for the flow of alleles from one parent to the other. Although what's absent here would be the F2s if we found them. So that's if these F1 hybrids were interbreeding, and we didn't see that, which does suggest some postzygotic isolation between them, something selecting against hybridization. And then to figure out the extent of hybridization between these species, what we can do is fit our data to Klein models. And so this is a Klein. What we're seeing here is on the x-axis, the geographic distance. So here's our westernmost site, that Palo Verde in Costa Rica, and our easternmost site, which is the Darien in Panama. And on the y-axis here is any kind of trait of interest that we can scale. And so here we've got what we're calling Q, which is admixture proportions for structure KO2, so 2 genetic clusters. And so that goes from a 1, Jacana spinosa, all the way to a 0, Jacana. And then in between them is where we see this hybrid zone. And so each locality here, this is an average of all those individuals in that locality. And this allows us to estimate the center and the width of the hybrid zone. So where it is, but also how wide it is. which can help us get an estimate of the strength of selection against hybridization. And so we'll look at some of these climbs later, but this is allowing us to start figuring out what's going on between these two species in the hybrid zone genetically. So the second question that we had, do the females of these species diverge in their competitive morphology and their behavior? And so in Jacanas, being heavier and more aggressive relates to how well you can defend and maintain your territory and then breed. And so between the two species, in alignment with our pattern of mitochondrial inheritance, we predicted that Jacana spinosa females should be larger both in competitive traits. So here that's the length of their wing spurs. These are like keratinous, kind of sharp fingernails that they have, but on the wrists, and their body mass. And we also asked about their aggressive behavior. And so to test this, we developed the first aggression assay in a shorebird, which is basically finding territorial females and using taxidermic mounts to test their aggression. And so I'll show you what our first prototype looked like. So we got rid of these leaves because they allowed the birds to become infested with ants. But basically that's what it looked like. And then we were able to connect a string around 20 meters away and hide behind a blind, and then test female aggression with these. These are all female-sized taxidermic mounts, and we were just testing conspecific. So we had 4 for Jacana spinosa and 4 for Jacana jacana, and then also 4 different vocal stimuli that we could then randomize and use different combinations. And so this was really exciting because it actually worked. It took like about a month to figure out in the field, but then these territorial birds really did attack. They got very close. They're kind of similar to what we think about with the sparrow aggression, but with Jacanas, there's a suite of behaviors that Emlyn described in his ethograms, things like pecks. I'll just look like that's a peck. This is like a wing spread. They do these like threats, and if you come to the party later tonight, you may see some of these same behaviors in our interpretive dance. And so basically, this is really exciting to get to work for a shorebird because it's usually done with songbirds with just song. But this moving and vocalizing helped us focus aggression on these mounts. And so what we found was that, as predicted, Jacana spinosa females had a more competitive morphological phenotype. So this means they had both a larger body mass and longer wing spurs than Jacana females. And we also found that, as predicted, Jacana spinosa females were more aggressive in response to a simulated territorial intrusion, and specifically that they approached these taxidermic mounts more closely than Jacana females. And so with this morphological and behavioral data in hand, I was then excited to go back to our clients and see what is the relationship between these competitive traits and our genome-wide markers. And so this is just a little bit of background about our predictions on clients. So we can look at the Klein center here. So this is for this genome-wide loci, and I already showed you what that Klein looks like for us. And then if we had asymmetric integration, we're going to find that the Klein center for this other trait of interest or locus of interest has moved across the hybrid zone and has a different center. And so this could potentially happen because if that locus or trait confers some kind of advantage, it could move across the population from one species to the other. And so even if these individuals are genetically more of this species, they have this locus or trait of the other. And so this is going along with this idea that genomes are semi-permeable and that different parts of the genome can move differentially. We can also look at the width of the Klein to infer things like the strength of selection against hybridization. And so a really narrow Klein suggests that this locus or trait is not widely moving between the species, and there's something limiting its introgression. In contrast, a really wide Klein, there might be weaker selection against that trait, or something that's actually promoting the introgression between the species. So we've already seen this. This is the structure admixture proportions for K of 2, which we're just calling the Q Klein or the genome-wide markers. And this gives us an estimate of where the genetic center and width of the hybrid zone are. And we mapped our first competitive female trait, female body mass. And this was another really exciting finding. So we actually see that its center here is distinct from our Q cline. And we can actually look at this in a different way. We can think of these like confidence intervals. They're 2 log likelihood values from our estimates of center and width. And what we're seeing is that they don't overlap. And so that's really exciting evidence of asymmetric integration. So this is, for us, kind of the first estimate that you can have asymmetric integration of a female competitive trait. We've seen other studies that look at male competitive traits or sexual signals, but this is indicating to us that in this context, female competition can work like male competition in other systems. And so the reason why we might be seeing this pattern is that in Jacanas, having a larger female body mass confers this reproductive benefit. And so individuals that have a larger body mass are better able to defend their territories and then breed on those territories. And so having a larger body mass confers a fitness advantage. And you can think about a scenario where hybridization between these 2 species allows the alleles underlying female body mass to go from Jacana spinosa into Jacana jacana. We looked at other competitive female. Oh, I should also say that we didn't find a difference in male body mass, so we weren't able to fit clines to that trait. We looked at another competitive trait, female wing spur, and found that its cline center did overlap with our genome-wide markers. So no pattern of asymmetric integration there, nor for male wing spur. But we also looked at these traits that are species specific. And so the facial ornament, right, there's a color difference there and also a shape difference. And this was really interesting that its cline was really closely tied with the genome-wide markers. And we found a similar pattern with the dorsal plumage going from brown into black. And so what this suggests is that these traits might play a role in reproductive isolation. They may be species-specific mating signals that could be important for keeping those 2 species separate. And I can explicitly test that by going back to Panama for 3 months this summer and actually doing these same aggression assays, but instead of with con-specific mounts, using heterospecific mounts and measuring the differences in response there. And so putting all of these clines together, in green here are our species-specific traits. In black is the genome-wide markers. And then in purple are our competitive traits. And one thing that we see is those clines where the competitive traits are much wider, whereas for the species-specific traits, they're more narrow. And so that can indicate that they may be playing a role in maintaining the species barriers, even though we could have some potentially leaky traits like female body mass that's introgressing asymmetrically. And so to conclude on this section, I found that the species are genetically distinct, and they do hybridize in that small region of overlap in Western Panama. I found that, as predicted, Jacana spinosa females have larger competitive traits and are more aggressive than Jacana jacana females. And this aligns with that pattern of the bias in mitochondrial inheritance. And then finally, that Jacana spinosa female competitive traits, well, just one in particular, female body mass, has asymmetrically introgressed in the hybrid zone. So in the process of my dissertation, I was really interested in behavioral divergence between these 2 lineages, specifically in aggression in the 2 species of Jacanas. And I wrote this proposal, this DDIG, to start thinking about what are the mechanisms underlying this divergence. And so this summer in Panama, I'll actually be going to look at gene expression in the brain and hormonal differences between females of these 2 species and between males and females within a species, to start getting an understanding of how this divergence can evolve. And I'm really excited to announce that I'm joining the lab of Kim Rosval for a postdoc to learn more about the evolution of female competition and the mechanisms that can lead to divergence in behavioral traits. And so I guess with that, I'd just like to thank everyone for coming and being a part of my PhD, and I'll take questions. Sorry. Yeah.
So you didn't have many back crosses, it looked like, not many F2s. So do you think how do you think that these traits are introgressing, given that there's not that much, that there seems to be strong post-psychotic reproductive isolation? Is there enough back crossing at low levels that these traits are able to move across?
That's a great question. I think that that could be explicitly modeled, and I haven't done that. But I think what it means is that there's got to be strong selection on body mass, and so I think we know that it confers a fitness advantage within a species. But I think, yeah, something considering how much gene flow you need for that trait to introgress to that extent. Yeah, but I think that could be.
So to what extent are advantageous traits like body mass contingent on plasticity? So you're sort of presupposing that it's a heritable trait.
Mm-hmm.
So it's modeled for progression expectations that it is by virtue of this, as opposed to say something that's environmentally.
Yeah, that's, I think, probably one of the major criticisms that I'm going to face in this paper that I'm working on. And so the reason I don't think it's environmental is because male body mass isn't changing. So I do think it's something that has a genetic heritable basis for females. It could be something, an interaction with hormones that I don't know about.
Could it be based on, say, a difference in sex-specific differences in time?
Yeah, I guess one thing that I didn't show here that might, again, speak against the plasticity is that for Jacana spinosa, the body mass actually doesn't change. So outside or inside the hybrid zone, those females have the same body mass. But then for Jacana, those females are smaller outside of the hybrid zone, and their mass increases in the hybrid zone. And so I think that speaks to there being something about being in contact with Jacana spinosa. It could be just selection for being in the presence of a better competitor, and there's maybe some kind of pre-existing genetic variation that's then selected upon, or it could be because of gene flow with that other species.
It sounds like this is a system that you're going to continue working on.
Yeah, yeah.
There's a whole series of things.
Mm-hmm. And we do know, for example, a study came out last year in chickens that showed a couple different loci that were correlated with larger body mass. And so that's something I'm really love to find, those loci in Jacanas.
You mentioned these interesting questions about F1s or hybrids may not be able to interbreed with other hybrids, or that the female matters in the sense that, sorry.
That's okay.
that only the Spinoza females interbreed with the Jacana females and not vice versa. I was wondering to what degree is it possible to do lab experiments on these birds to actually explicitly test these?
Yeah, that would be a dream, to have some kind of captive colony where I could actually do these crosses. But so this bias and inheritance of mitochondrial DNA, it can indicate either that female competition is driving the mating patterns, and or that there's something with the other cross not being viable. And so I actually have some new data that I didn't show, where Jacanas, there's a back cross that has a mitochondrial haplotype of Jacana. So it can happen. Just because it can happen, it could still be like less effective or more rare, either because of this competition hypothesis or the hybrid inviability. So because I can't yet do these dream captive crossing experiments, I can actually still start looking at post-psychotic isolation by looking at, for example, Z-linked loci, which other people have shown when they have narrower clines, that can be indicative of selection against hybridization. And I can, I guess, still try to figure out, there's also now we know loci that are associated with female sterility. And so I can look at the hybrids and see, I guess, if they have those loci or some pattern that indicates a different parental cross affecting that sterility. Those are all good future steps.
I think that we should thank Sarah. This is 10. Thanks, guys. Well done.
Good.
Yeah, it's ongoing. A lot of greenhouse work. Trying to get her to write this experiment. A lot. But yeah, I was working with Daisy yesterday. She got very great. She's got on her computer now, so she's an independent agent.
Halfway through, it's like, oh, I'm literally finished right now. Yeah.
Sure.
Okay, if I go ahead and grab this.
Yeah.
Thanks for watching, everybody.